So the next talk is by Professor Anandamohan Ghosh from IISER Kolkata. And Ananda, if you are there, can you start sharing?
I'm trying to. Can you hear me?
Yes, we can.
I'm trying to share this screen.
So,
can you see it now?
Yes. Can you make it full screen?
Okay.
Okay. Yes. Over to you.
Okay. So thanks, Una, and good morning, everybody. So I should first thank Tridib for making this impossible possible by convincing Deepak and Ms. Tansi to let this meeting happen, which was due for a very, very long time. Okay, so I'll talk about first passage time in gene regulation. And so this is a very broad title as it seems, but I will actually do a very specific problem and I will explain it as I go along. So the key player in the molecular biology is this gene regulation, and which is a two-step process where all the information in a DNA is transcribed to a single strand mRNA, and this mRNA translates into proteins, which are the building blocks and the functional units of all cellular processes. So why this problem is interesting, because with the very nice experimental techniques developing over the last couple of decades, so earlier people used to think that this is a generalization of a birth-death process. So all the protein distributions or the mRNA distributions will follow something like a Poisson distribution, but that is not the case. If one goes into single cell and measure the mRNAs and proteins by tagging them with fluorescent particles as a function of time, one can see that they are produced in bursts. So since this mRNA and the protein synthesis involves a binding of a lot of external factors, so this is inherently noisy and as well as bursty. And what I'm going to tell today, how these processes can be studied in the context of a first passage problem. And this is also interesting because not only the proteins need to be produced in a certain number, because until it reaches a critical threshold, it cannot trigger a downstream process. And that too, since many processes are taking place simultaneously, so there needs to be certain order of synchronization among them. So each process also needs to synthesize protein at a sufficient number and at a very precise time. So in that context, the first passage time is a very relevant thing to study in the context of gene regulation. So there can be, as you can imagine, gene regulation can be a very complex network of many proteins and stemming out from the gene. But here I will be basically focusing on three simple motifs. So let's call the first one as a non-regulation, because here the gene is always on and it transcribes into an mRNA and it translates into a protein. On the other hand, there can be transcriptional regulation where there's due to RNA polymerase binding, which carries out this transcription only for the active gene. So the gene can switch with certain rates between off state and the on state. Okay, so here all this regulation is taking place at the gene level. So this is called transcription regulation. Or it can be also happen, the regulation happens at the post-transcriptional level, but there are small tiny molecules called small RNA, which enables this mRNA to switch between an active and a deactive states. And if it's an active state, then only the ribosome can bind and the proteins can be synthesized. So we'll be comparing first passage times of these three templates. So what is the general prescriptions? I won't go through all the details, but I will give you the general algorithm. So we start with this reaction network, and let's associate M with the mRNAs, small n with the proteins. And we built up a master equation where for the probability that M mRNAs and N proteins are synthesized at say time t. Okay, so it's a fair amount of bookkeeping. One can write a master equation. Then the algorithm is that we make the equations dimensionless. We use a generating function to write a partial differential equation, which in some cases, in some limits can be solved for the protein distributions. Like in all the models that we study will be solved in the limit that gamma is much greater than 1, which is the ratio between the relative degradation rate of the mRNAs and protein. and which is a reasonable thing or to assume, because proteins are much stabler and in comparison to the mRNAs. So in this limit, this protein distribution can be obtained. And what we'll also need in the next slides is that we can get the exact expression for the evolution of the mean and the variance of the proteins. So this is so as in the large time limit. So this mean converges to the steady state value, and which is not surprising because more the Km, Kp are the translation and the transcription rates, and more is Km, Kp, the yield is more. And again, if I look at the protein fluctuations, so since this xi factor is always greater than 1, or is always a positive number, so this variance of the protein numbers is always greater than the mean. So this final factor will be 1. So process is intrinsically worst. So even for this is a two-stage model, which is also a other name of the no regulation or our control case. And if we introduce a switching of the genes between the on and the off state, the same process can be followed. Only here we distinguish the probabilities into two categories depending on the on state and the off state. Similarly, PDs can be written, and by methods of characteristics, the probability distributions can be solved. And again, if you look at it, the mean and the variance of the proteins follow a similar kind of evolution. Only here, the steady states will change because it is depending on the fraction of the on states, it will be accordingly altered, and the burst parameter will also differ. Okay, so otherwise the calculations is almost identical. What we also did for the post-transcriptional one, where there is only one single small RNA is involved. So again, even in this case, by introducing a genetic function, we can again have a complicated looking partial PDE, which again can be solved for the large gamma limit. Again, I would like to draw your attention, the mean and the variance are still of the same form, and the steady states values and the burst parameter changes accordingly. Now, coming to our concern to calculate the first passes time, one could have gone the traditional way of solving the master equation, like casting it as a backward Fokker-Planck equation and calculating the cumulative distribution and taking the derivative of that with the first passes time distributions. But that route becomes complicated and often impossible in these problems. So we have done a very approximate calculation, and with approximation, we also pay a price that we can only get a very qualitative behavior. So the idea is the follow, that if we take this blue curve, which is the evolution of the mean of the proteins, and if it hits the threshold value at some time t, so that is our mean first passes time. And since the growth is exponential, this can be approximately calculated. And then on this mean curve, the blue curve is flanked by the red curves, which are the variances, how the variances evolve. And if we assume that the t is sufficiently large than that such that TL and TR are small, so we can expand this variance for a small TL and TL as a Taylor series. And our variance of the first passes time will be the average of this TR and TL. And the idea is that we can write this thing as a derivative or as a function of the mean and the variance of the proteins, which we have calculated in the previous slide. Okay, so using this approximation, if I put together all the results that we have accumulated over the years, that is in these three scenarios, these are the steady states, these are the protein burst sizes, and from which we can calculate the first passes time where standard deviation, first passes time mean, and calculate the FPT noise, okay, which let's call that thing chi-square, and which is 1 over steady state value and proportional to the burst size. Okay, now to compare all the three models, then we kept the steady state fixed for all the three models. See, if NSS is our fixed, then it is not too difficult to see that the burst size of the transcriptional regulation will be greater than our control, and the burst size of the post-transcriptional regulation will be least. And so in other words, it will mean that the first passes time noise will be the least for the post-transcriptional regulation. Okay, so this was exciting. That means the small RNA induced post-transcriptional regulation probably reduces the equity noise, which can be advantageous for the cell. But then it was not quite encouraging, because if we go to the KP-Km plane, and this is in genetics, this is also called the crick space. So if I take KP-Km constant, that is the constant protein abundance. Okay, so all the dashed lines are for the constant protein values, and if I KP-Km both changes, then along the blue curve the yield changes, or the steady state value changes. But if I increase Kp, then I can reduce this first basis time noise. So that means, let's assume that increase the precision or the timing precision of the regulatory mechanism. But at the same time, to keep that yield constant, my Km has to be larger. Km has to be larger, and in process of doing so, my transcriptional cost will increase. So in this cartoon, so you can.
You have 2 more minutes.
Okay, thanks. So post-transcriptional regulation will make the process precise in sense of timing, but it will also increase the transcriptional cost. So then we realize that considering a single small RNA may not be a good idea, and the small RNAs are very clever, so they always work in a group. And if we include more such small RNAs, so in this cartoon I just showed 2 of them. I haven't shown the calculations, but the idea is that since introducing 2 small RNAs essentially introduces more degrees of freedom, so protein mean and the variances can be intelligently controlled, and in this case much desired precision and keeping the same economic in the transcriptional cost can be achieved. So there are approximations. So but anyway, so this is a very interesting area where much nice work can be done. And in the context of the lysogenic cycle, recently Dibyendu has done a very nice calculations, I mean, of the first passage time problem. So, and there are many other aspects one can look into. So most of this work was done by Kuhile, who is now my PhD student, who is now a postdoc at Technion. And most of this work was initiated by Mayank and Anudeep, who are almost completing their PhDs at various other universities. And finally, a big thanks to Deepak and Mustansir for very nice interactions, and which will be very fondly remembered. And thank you all for your kind attention.
Thank you, Professor Ghosh. So there are many questions I see. So Dibyendu, you can go first.
Yeah. So I just had one question. That is for the transcriptional regulation, the biophysical processes that at least I sort of have come across is like cell lysis, cell division, and all that. So for this post-transcriptional regulation, what sort of biophysical processes do you have in mind with the small RNAs?
So small RNAs essentially do is that they bind with the mRNAs, and if they do so, then only the ribosome can bind. So this cooperative phenomena then initiates the translation process.
But at some cellular scale, is there any sort of phenomena you had in mind for this post-transcriptional regulation that sort of could be serving as a motivation, a biophysical motivation, do you think? But I'm just wanting to know the literature. I'm not familiar with that. That's why.
So these small RNAs are slightly recent, and they have been, discovered this genetics in the various problems. So often I cannot give you one single example, but there are various instances where they play a very important role.
Okay, thank you. Yeah.
Next question is from Professor Debashish Choudhury. You can go ahead.
Anandamohan, very nice talk. I have one question, because you compared also 3 models. Did you try to at least capture some experimental situation related to actually the Bender question, taking some known parameters and to see which one fits better, or can you throw light on some real particular case, at least some case? Yeah.
So these parameters that we have chosen are taken from literature. So we didn't collaborate with experimentalists directly. But this kind of things, like in Dibyendu's recent work, he has actually worked on the experimental data. So there he has actually fitted to this. I guess the protein was wholene, right? So the distribution of this fast passage time for the wholene protein matches exactly with the experiments. So it can be done in appropriate data. It can be fitted to any experimental data.
No, but so did you try to see,
No, so we just take the data from what is available in the literature. So when we choose these parameters, it's taken from the literature.
No, so that is the input. I'm talking about the output. That means you have some prediction. Did you try to see, how good the predictions are?
I don't see that anybody has measured the fast passage time apart from this Dibyendu's recent paper in experimental situations. So I don't know what to compare with it directly.
There is one person, Abhyuday Singh. I'm sure you're familiar. So Abhyuday Singh has tried in some context to compare, maybe something of that kind.
Devashish, actually their data only we collaborated and we actually matched. So, but that is not in the context of small.
No, I understand. So I'm saying
so that is why I was curious. That is there more experimental things that
exactly. I mean, something similar. I was
but we collaborated with Abhyuday actually.
That's okay.
Okay. Thank you. So there is one more question in the chat and one comment. So Shantanu Kadam wants to know whether FPT can be done on cellular signaling networks and if there are any limitations on that.
So the limitations that we have is that we assume that this gamma to be large, so which in some is not very generic. So by taking this gamma to be large, we in this approximate calculations, we in some way say that the fluctuations are not overwhelming, so that this approximation goes through. Yes, so in principle one can do it, but one has to be aware of the limitations of the approximations.
So let's thank Professor Ghosh once more for this great talk, and let's move on to the next one.